Hey everyone, and welcome back to The Explainer. I am absolutely thrilled you're here today because honestly, we are going to explore one of the most elegant, highly efficient biological factories known to science, your own cellular metabolism. Specifically, we're zeroing in on carbohydrate metabolism and glycolysis. If you've ever wondered exactly how your body keeps the lights on, well, you are in the exact right place. Just to put things in perspective, did you know your brain, which is only about 2% of your body weight, actually consumes around 20% of your daily glucose? It's incredibly demanding, so today we're going to see exactly how it gets fueled. All right, here's our quick roadmap for today. We'll hit the basics of metabolism, figure out how glucose gets into our cells, break down glycolysis into its energy investment and payoff phases, and finally look at how the whole thing is regulated. So I want you to just think about this for a second. How does the raw material of a simple morning meal, a piece of toast or an apple, actually transform into the exact molecular currency needed to keep you alive, moving, and thinking? It doesn't just happen by magic, right? It's a meticulously controlled biochemical assembly line. And today, we're walking the factory floor. Okay, let's jump into section 1, the basics of metabolism, looking at catabolism versus anabolism. Before we really get into the weeds with glycolysis, we kind of need to understand the overarching business cycle of the cell. It's this constant balancing act between breaking things down and building things up. You can think of metabolism as two opposing but totally complementary forces. First, you've got catabolism. This is the breakdown of complex, large molecules to produce energy and simpler compounds. It's literally like spending your raw materials to generate power, like breaking down stored glycogen in your muscles into usable glucose. On the flip side, we have anabolism. This is the synthesis of complex molecules from simpler ones, which actually costs energy. This is your cells saving up and building for the future, like taking excess glucose and storing it away in the liver. To really visualize this, imagine a catabolic reaction where an enzyme utilizes water to just tear apart a complex molecule, releasing a burst of energy in the process. Then, in an anabolic reaction, the exact opposite is happening. An enzyme is actively consuming energy and releasing water to stitch simple, small molecules back together into a complex structure. Our main event today, glycolysis, is a textbook catabolic pathway. We are tearing apart a sugar molecule to extract its energy. Moving right along to section 2, getting glucose into cells, or what I like to call the factory front door. So before our cellular factory can actually process any raw materials, we need a way to transport glucose from the bloodstream right through the front doors of the cell. And glucose can't just waltz through the lipid cell membrane on its own. It needs help. Our body uses these specialized doors called GLUT transporters. Think of them like highly exclusive bouncers at different clubs all around your body, each with their own strict entry requirements. GLUT1 is heavily used by your brain and red blood cells for steady baseline entry. GLUT2 handles the high-volume traffic over in the liver and the beta cells of your pancreas. GLUT3 is an extra door specifically for neurons, while GLUT5 is a specialized bouncer that strictly lets fructose in. But the one I really want you to focus on is GLUT4, which guards muscle and adipose or fat tissue. This right here is a super important clinical connection. For muscle and fat tissue, these GLUT4 transporters aren't just sitting open on the outside of the cell. No, they're stored away in little vesicles deep inside. Insulin is the essential VIP key here. When insulin binds to the cell, it triggers those GLUT4 storage vesicles to move right up to the plasma membrane, opening the doors and dramatically increasing glucose uptake. Without insulin, that glucose is just stuck outside. This is exactly why insulin resistance in conditions like type 2 diabetes is such a massive problem. The key doesn't work, the bouncers stay hidden, and the factory literally starves while glucose just builds up in the blood. All right, on to section 3, glycolysis, specifically the energy investment phase. Now that our glucose is successfully inside the factory, well, just like any startup business, we have to spend some of our own capital upfront if we want to make a profit later. The entire blueprint of this process is neatly divided into two halves, the preparation phase and the payoff phase. This is a 10-step enzyme-catalyzed pathway that happens strictly in the cytosol of the cell, so outside the mitochondria. And the overall goal? It meticulously converts a single 6-carbon glucose molecule into 2 3-carbon pyruvate molecules. Let's look at how that first half actually works. So, diving into that energy investment phase, steps 1 through 5. Right away in step 1, the enzyme hexokinase takes an ATP molecule and uses it to phosphorylate glucose into glucose 6-phosphate. This is crucial because it effectively traps the glucose inside the cell. Then, after a quick shape shift and isomerization into fructose 6-phosphate, we hit step 3. Here, the enzyme phosphofructokinase 1, or PFK1, burns a second molecule of ATP. At this point, our cellular startup is officially operating in the red. Finally, the enzyme aldolase comes in and cleaves this newly modified molecule right down the middle. After a bit of rearranging, we're left with 2 separate 3-carbon molecules called glyceraldehyde 3-phosphate, or G3P. We primed it, we spent our cash, and we split it in half. Which brings up a pretty obvious business question, right? We've just consumed 2 whole ATP molecules. We are in a serious cellular deficit. So, when does the cell actually see a return on this investment? And that brings us to section 4, the energy payoff phase of glycolysis. This is the moment our factory finally starts harvesting the energy locked inside those chemical bonds. It's a huge turning point. We take those 2 G3P molecules we just made and send them down the second half of the assembly line, steps 5 through 10. And here is the absolutely crucial thing to remember. Because we have 2 G3P molecules from that original split glucose, everything that happens from this point forward happens twice. The factory is running 2 simultaneous assembly lines, and that is the real secret to making a profit here. Let's break down how we actually get paid. In step 6, G3P dehydrogenase oxidizes our molecule, giving us our first high-energy intermediate, NADH. Think of NADH as a tiny molecular taxicab that picks up high-energy electrons and ferries them off to be used later. Then, in step 7, the enzyme phosphoglycerate kinase performs something called substrate-level phosphorylation. That's just a fancy way of saying an enzyme is directly handing a phosphate over to ADP to create a brand new ATP molecule. Awesome! We do some more molecular rearrangement in steps 8 and 9 to form phosphoenol pyruvate, or PEP, which is highly energetic and very unstable. Finally, in step 10, the enzyme pyruvate kinase transfers a phosphate from PEP to ADP, generating our final ATP, and leaving us with our end product, pyruvate. 2. That is the magic number. Because we ran that payoff phase twice, once for each of the 2 assembly lines, we produced 4 ATP and 2 NADH taxicabs in total. But remember, we spent 2 ATP back in the investment phase. So if we do the math, the ultimate return on investment per single glucose molecule is exactly 2 pyruvate, 2 net ATP, and 2 NADH. This is literally the foundational math of your cellular survival. Which perfectly sets up our final section, section 5, regulation and final pathways. Look, an efficient factory can't just run blindly at maximum speed 24/7. It needs quality control, management checkpoints, and a solid plan for its final products. Out of the 10 steps of glycolysis, 7 of them are totally reversible. They just flow back and forth based on supply and demand. But 3 of them are strictly one-way streets. For anyone studying this, you absolutely must memorize these 3 irreversible checkpoints. Hexokinase at step 1, phosphofructokinase 1 at step 3, and pyruvate kinase at step 10. Why are they one way? Because the cell commits major energy resources at these exact points, and it can't turn back without using completely different specialized bypass enzymes. PFK1 in step 3 is the really big one. It acts as the primary rate-limiting step for the entire operation. It's essentially the master dial controlling how fast the whole factory runs. Finally, we have to talk about what happens to that final product, pyruvate. It has 2 diverging fates. If oxygen is present, which we call aerobic glycolysis, that pyruvate and the NADH taxicabs are sent straight into the mitochondria. The NADH uses special transport shuttles, like the malate aspartate or glycerol phosphate shuttles, to get inside, leading to complete oxidation and a massive ATP yield. But what if there is no oxygen? Think about that deep physical burn in your muscles during an exhausting, heavy workout. That burn is your cell switching to anaerobic glycolysis. It uses an enzyme called lactate dehydrogenase to convert pyruvate into lactate. Why would it do that? Because this reaction quickly regenerates empty NAD plus taxicabs, which you absolutely need to keep the glycolysis assembly line running. This way, the cell can at least keep generating that small, rapid stream of 2 ATP per glucose to keep you moving in an emergency. And honestly, that just highlights the sheer, beautiful adaptability of human metabolism. We've got this incredible primary aerobic engine, but we also have a vital, rapid-fire anaerobic backup generator, which leaves us with a pretty fascinating thought to close out today's explainer. If anaerobic glycolysis is only yielding a meager 2 net ATP compared to the massive payout of the mitochondria, just how long can your tissue survive on this emergency backup when pushed beyond the limits of oxygen? It is an incredible testament to cellular survival, and definitely something to ponder. Until next time, keep learning, stay curious, and I'll see you in the next explainer.